So, Bismillahirrahmanirrahim. In this video, we will discuss glycolysis, which has been divided into two reactions, that is, energy requiring phase and energy generating phase. But in this video, basically, we will discuss the energy requiring phase, that is the first phase, with all its reactions, all its enzymes, and all the type of things that are happening, it's all its regulations. So, first step is the conversion of glucose into glucose 6 phosphate, that is now, since a phosphate group has been added to glucose at the sixth position, ATP is used and ATP is added. And when phosphate group from ATP is removed, what is left behind? ADP. So, ATP is added, ADP is removed, and glucose 6 phosphate is produced with phosphate attached at the sixth position. Now we have hexokinase or glucokinase. Both of these both of these enzymes, they catalyze the conversion of glucose into glucose 6 phosphate. Now, firstly, we will look at the type of hexokinases. They are hexokinase number 1 to 3 and hexokinase number 4. So hexokinase number 1 to 3, they are found in most tissues. They are inhibited by glucose 6 phosphate. We will discuss inhibition later. And they have low Km, low Michaelis Menten constant. So they have high affinity for glucose and they can be activated by small amount of glucose. They have low Vmax, so they cannot trap the glucose inside the cell, and they can catalyze this first step, the glucose to glucose 6 phosphate, in several hexoses, in glucose plus several hexoses. But now this hexokinase 4, it is mostly called glucokinase. It is present in liver and pancreatic beta cells. Both the Michaelis Menten constant and Vmax, it are more in these. So increased Km means low affinity, so it requires large amount of glucose to be activated, hence it acts as a glucose sensor. And increased amount of Vmax, it means that it can trap glucose inside the cell. So when it can trap glucose inside the cell, it will not allow the glucose to move out of the cell, and hence it can be used to treat hyperglycemia. And it has a key role in adrenergic response to hypoglycemia. Now, what happens? Maturity onset diabetes type 2. What is this type of maturity onset diabetes type 2? Um, in this reaction, majorly insulin secretion is impaired and there is hyperglycemia. Why does it happen so? Because there are mutation in this hexokinase 4 or glucokinase. Now we will look at the regulation of this hexokinase 4 or glucokinase. Let's look. For its regulation, we have this step. So as I told you, glucose was taken into the cell by, it can be done by the sodium dependent ones, can be done by sodium independent ones. So glucose is now inside the cell, and glucose has been converted into glucose 6 phosphate by this enzyme glucokinase. This glucokinase is activated by this glucose. Whenever we have large amount of glucose, the glucokinase will act on it and convert it into glucose 6 phosphate. But what will it do? Firstly, it will separate it from the glucokinase regulatory protein, which is present in liver cell nucleus. And whenever glucose is large amount, it's present in large amount, what it will do, it will separate this glucokinase from its regulatory protein, and hence more amount of glucokinase will be present, it will be converted into glucose 6 phosphate. But now whenever fructose 6 phosphate is more in amount, it will cause the glucokinase to move inside the cell and be attached to its glucokinase regulatory protein. So glucose activates glucokinase by removing it from the glucokinase regulatory protein, whereas fructose 6 phosphate inhibits it by causing the glucokinase to be attached to its regulatory protein and not be available for the conversion. Activated by glucose, inactivated by fructose 6 phosphate. So now we are done with the first step. Now we will go to the second step. The second step is conversion of glucose 6 phosphate into fructose 6 phosphate by phosphoglucoisomerase. This phosphoglucose isomerase is, you can say, isomerizing type of enzyme, and this reaction is reversible. It can run in both directions, but this one was irreversible. Now the third step is fructose 6 phosphate being converted into fructose 1,6 bisphosphate. So what happened over here is that at the position number 1, additional to the position number 6, we got another phosphate added. Where does this phosphate come from? ATP. So you will add ATP, remove to the ADP form, you will get fructose 1,6 bisphosphate. Now this reaction is catalyzed by phosphofructokinase 1. Now this phosphofructokinase 1 and its regulation is very important because this is the rate limiting or regulatory step. So we will move to this phosphofructokinase 1 regulation. So now it is regulated by both the levels of ATP and by fructose 2,6 bisphosphate. So now whenever ATP levels are high or whenever citrate levels are high, they are going to inhibit the fructose phosphofructokinase 1. So whenever ATP levels are high, citrate levels are high, they are going to inhibit phosphofructokinase 1. But whenever ATP levels are low or we have more ADP or AMP levels, it will activate phosphofructokinase 1. Low levels of ATP, activation. High levels of ATP, high level of citrate, inhibition. Now there's another thing which is fructose 2,6 bisphosphate. Now this fructose 2,6 bisphosphate has positive regulation. Whenever it will be more in amount, it will cause this phosphofructokinase to be activated and hence catalyze this reaction. Now this fructose 2,6 bisphosphate is itself made by phosphofructokinase 2. Now this phosphofructokinase 2 is very important. This phosphofructokinase 2 is very important because it is a bifunctional enzyme. It is having two domains. One is the regulatory one is the kinases domain, other one is the phosphorylases domain. So now whenever phosphate group is going to bind to this phosphofructokinase enzyme, what will happen? Phosphorylation by phosphatase group, it will cause its inactivation. So what do I mean by that? I said that it has two domains. One is kinase activity, one is phosphorylase activity. So whenever there is like glucagon present, the glucagon means low amount of glucose. That glucagon will cause its phosphorylation and activation of its phosphatase domain. Whenever its phosphatase domain is activated, what will happen? Phosphate group will attach to it and fructose 2,6 bisphosphate will not be produced. When fructose 2,6 bisphosphate is not produced, it means that the activation process that this was going to the PFK1 phosphofructokinase 1 is not present and hence glycolysis will be stopped. But when we have large amount of glucose, it means that we want this glucose to be broken down. We want glycolysis to happen. So this glucose will cause the production of insulin. This insulin will go to this phosphofructokinase 2 and activates its kinase domain. You can remember the mnemonic I know or IK. So insulin supports the kinase domain. Kinase will cause the fructose 2,6 bisphosphate to be produced in more amount. When this the fructose 2,6 bisphosphate is more in amount, what will it do? It will do the positive activation and hence we will have more amount of phosphofructokinase 1 activated. When it is activated in more amount, what will happen? Increased glycolysis or increased conversion of fructose 6 phosphate into fructose 1,6 bisphosphate. I hope you understood this phenomena. Low glucose, we don't want glycolysis to occur. Hence, phosphatases will be activated. High glucose, we want glycolysis to occur. So, insulin is going to activate kinases and kinases will cause more fructose 2,6 bisphosphate and hence more amount of activation and hence more conversion or more glycolysis. So, you can remember by this. So, this is the same thing. What is well-fed state? When you have more glucose, when you just took a carbohydrate-rich meal, that glucose causes increased insulin. That insulin causes kinase activity and PFK2 is activated. Fructose 2,6 bisphosphate is more and hence more PFK1 activation. Similarly, in fasting, you don't have more glucose, you have less glucose. You don't want glycolysis to occur. Hence, glucagon is now more. This glucagon activates phosphorylation, causes phosphorylation through phosphorylase. PFK2 is phosphorylated it is inactivated, or you can say it does not allow fructose 2,6 bisphosphate to be produced, and hence PFK1 is also inactivated. Now, the so up till now we are done with first step, glucose to glucose 6 phosphate by glucokinase. That was activated by glucose itself. The arrow is wrong. That was activated by glucose itself, and it was inactivated by fructose 6 phosphate. Now, this glucose 6 phosphate gets converted to fructose 6 phosphate by phosphoglucoisomerase. Done with this, fructose 6 phosphate to fructose 1,6 bisphosphate, activated by fructose 2,6 phosphate and low amounts of energy, but inactivated by high amounts of energy, high amounts of citrate. Now, this fructose 1,6 bisphosphate, in the next step, it is broken down into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate by the enzyme aldolases. Now, this aldolases enzyme, it is not regulated. One thing that we found in the book about it was the aldolase B, which is its hepatic isoform. It has the ability to cleave fructose 1 phosphate, but this aldolase that we are discussing right now, it has the activity to cleave fructose 1,6 bisphosphate 1. Now, in the next step, what we need the glyceraldehyde 3 phosphate to be more in number. So what will we do? We will use an enzyme, triose phosphate isomerase. So it has the ability to convert either glyceraldehyde 3 phosphate into this form, or this one into this form. So what will majorly happen, that this dihydroxyacetone phosphate will be now isomerized. It will be converted into glyceraldehyde 3 phosphate. We will get two molecules of glyceraldehyde 3 phosphate, and hence the whole step is catalyzed by triose phosphate isomerase. With this, we are now done with our 5 first reactions of the energy requiring process. 2 ATPs are used in this reaction, this first at the first step, and second ATP was being used in this process, that is fructose 6 phosphate into fructose 1,6 bisphosphate. I hope you understood my whole um, biochemistry about the energy requiring phase. And if you did, if you like all the mnemonics, please like, share and subscribe to my channel, because in the next video we will discuss this energy generating phase, inshallah, and all its regulation and everything related to it. I hope you understood each and everything about the energy requiring phase of glycolysis. Let's meet in the next video, inshallah.